Hi, this is a re-recording of my live talk from Monday, which unfortunately got truncated, for those who would like to watch it. Welcome to my talk. My name is Michael Grau and I'm researching as a postdoc at the University Hospital in Münster. My background is actually not medicine, but I'm coming from the complex systems group of the University of Marburg in physics. Now let me explain you my method, signal dissection by correlation maximization, Sdcm, which yields signatures of nonlinear interactions in high-dimensional complex data sets, for example, transcriptome measurements. First, let me introduce you to the problem we are trying to solve. You see a microscope image of cancer cells, and of course you know they show no natural cell death and proliferate uncontrolled in the body, and we need to understand it in order to be able to stop it. And for this, we measure on much more granular molecular level, not just on microscope level, over 55,000 transcripts. And the typical data set for about 500 primary tumor samples looks like this, and you have to zoom in quite a bit before you can see individual measurement values. To give you a familiar number, it's about 27 megapixels, but in contrast to your typical photography, it doesn't have any neighborhood information. It's a quite complex signal, also with system random noise, and the complexity stems from the fact that you effectively measure a net sum of many overlapping effects, many different cell types per sample, and many different concurrent gene expression programs per cell. And in this sum, we would like to find signatures of interactions. Let me give you an example of an important signature for diffuse large B-cell lymphoma, which is a disease where, unfortunately, there are alone in Germany 5,000 new cases each year. And if untreated, it ends deadly. Already in 2000, Alizadeh and others found a pattern by hierarchical clustering, and by comparing this with healthy activated or germinal center B cells, they distilled a signature which can be used to sort patients, as done here by Visco and others in 2011, from ABC-like to GCB-like DLBCL. And the important bit about this gene expression signature is that it's clinically highly relevant because the 5-year survival of ABC patients is only 40%, whereas GCB patients survive on average about 70%. So it's a better surviving subtype. And we can learn from heterogeneity in the data, and ultimately we want to learn from this heterogeneity how to get more specific therapies for patients. But recall, the data is huge, and you first have to find such signatures. Which brings me to my method, signal dissection by correlation maximization. If you want to know all the details, you find the citation down there. We published it in Nature Communications 2019. It contains a lot of math, so maybe it's easier to see me tomorrow, Thursday, in my poster session and ask questions there. In this 10-minute talk, I can only give you an overview over the key concepts. Let's start with the signal model. That's straightforward. The heat map you saw earlier, the matrix M, is dissected and represented as a sum of matrices EK. Each EK is a signature plus a noise matrix eta. And our task is to reverse the sum into summons. And of course, this is a mathematically highly ambiguous problem, so we need additional constraints. And these constraints need to be sufficient for dissection, but should be as few as possible in order to fit as broad as possible range of natural phenomena. And my idea was to use a consistency which is defined as follows. For an ordered subset of genes, which is initially unknown, and for an initially unknown ordered subset of samples, I demand bimonotonic consistency. Sounds complicated, but essentially only means that for the gene rows and the sample columns, the EK is in each row and each column monotonic. And all other rows that do not participate in this signature are zero. Okay, the problem now is we need to find in a huge 2 to the power of n subspace these subsets and these suborderings. Let me now explain you the main concepts of Sdcm with a simple three-dimensional concept example of only 3 genes X, Y, and Z. The colors are just for you, the method only sees the sample points. The first step is to select an initial representative that was actually measured, giving an initial axis in the gene space. Second step is to optimize this with additional representatives that are nearby. The third step is to apply bimonotonic consistency, which yields this yellow monotonic curve. And the fourth step is to dissect those parts of the sequence that can be explained by it. Again, with a bit more details. The first step is to select an initial representative that maximizes a signature functional. And the signature functional is the higher, the more points are consistent with this direction. And just one representative is never really good, so we optimize it by locally maximizing the signature functional, which gives me this initial yellow line. And nearby is defined by weighted cosine distance, which corresponds to weight isosurfaces in forms of double cones. And within this, we call it signature focus, the bimonotonic regression takes place, which then yields again a monotonic curve. And to connect this to the heat map view, let's view all the points in their coordinates, X, Y, and Z. And each column is a point. And the gray columns are just those points that are outside the signature focus. And the bimonotonic regression, the yellow curve, is actually equivalent to the matrix view of the blue and red coordinates down there. And it's the same in high dimensions, just more than 3 rows. Okay, then we can dissect it again and iterate for the remaining effects and this offset effect. And at this time, a statistical model for correlation significance and signature strength significance says nothing is significant anymore, and Sdcm terminates. Here you see all 4 signature curves that were detected again. And of course, in the paper, we also simulated high-dimensional versatility tests and compared with a bunch of other methods, including principal component analysis, independent component analysis, non-negative matrix factorization, hierarchical clustering, a biclustering method called FEBS and FEBS, and a bunch of other biclustering methods. And from all these methods, PCA showed the second best sensitivity and specificity on the simulated datasets after Sdcm. So let's compare these 2 methods a bit more on a conceptual level. Here you see the 3 principal components of this dataset, and PCs look in the direction of maximum data variance, whereas Sdcm looks for directions of maximum consistency or correlation. PCs are linear, whereas Sdcm signatures are monotonic curves. And PC uses full projection as reduction step, causing all principal components to be orthogonal to each other, whereas Sdcm only dissects the explained signal parts within the signature focus. And if you recall what Andre said on Monday with respect to explainable AI, I think that this model for a suitable broad range of nonlinear datasets is more explainable than principal components. Alone in this 3D example, you can see that the PCs essentially point into empty space and mix several of those simulated source effects if you project on them. This brings me to a few results from Sdcm for real data for diffuse large B-cell lymphoma gene expression. And here you see those 2 Sdcm signatures that were validated with independent survival data to be the most predictive of all signatures, including principal components, independent components, and what the other comparison methods yielded. And a bivariate Cox model based on these 2 signatures was able to predict survival differences best. First, we validated these 2 survival signatures on purely gene expression level in 2 independent patient cohorts, one microarray based, one RNA-seq based. And as you can see, they exist there too. And the interesting bit is that if you look on the patient subgroup that is on the right side of each of the two signatures, and then do the Kaplan-Meier analysis and show it within the known GCBDL-BCL subtype, which is the subtype known to have better average survival, you find a subtype of essentially of a significantly inferior survival, which was surprising because GCBDL-BCL is usually the one who was thought to have better survival. Okay, this brings me to an outlook. Sdcm is a mathematically general method like PCA. It can be applied to essentially any features cross samples input matrix. Here, a student team of mine applied it to beat AML data from the DREAM challenge this spring, and we made it up there. The model is only based on the Sdcm signatures and gene expression data. You have to know that in the competition there were also TP53 and other mutational data available, but due to deadline reasons that was not part of our model. Otherwise, maybe we would have been up there with the other four. Another students team applied Sdcm to gliomas and found heterogeneity there and dissected it there. And for time reasons, like on Monday, I will skip over the two physical examples and come to the future. We are looking into pan-cancer heterogeneity now with all the difficulties with lab effects. And we would really like to integrate Sdcm, which is like PCA matrix level method, with prior knowledge based on networks. And two of our PhD students will work on this in this direction in the next 3 years. And especially in this context, we are also open to inspiration and maybe collaboration. If you want to try Sdcm, you can find its source code on GitHub. And I'd love if you'd like to follow me on Twitter. There you will also get news about new versions of Sdcm. And let me close by thanking my current director, my PhD advisor, and my current team of PhD and master students. And of course, I thank you for the attention. Was a bit more tired than on Monday in a live talk, but I hope you enjoyed it. See you.